Hello, today we are talking all about proteins. This should be a review of what you've already learned. This is not it's not actually its own subchapter in chapter 7. This is sort of a review of what you've learned throughout your entire science career, but we're going to focus in on what you should know and be able to apply to the proteins being created through protein synthesis. So, let's just give a general definition. This will be our working definition of what a protein is. They are stable, folded 3D structures of polypeptides. Polypeptides are the chain of amino acids that fold into proteins. And for what we're focusing on, there are 4 levels of protein structure. You should be able to differentiate between the 4 levels themselves. The 4 levels are primary, secondary, tertiary, and quaternary. Primary structure is the amino acid sequence. They are connected via peptide bonds. A secondary structure is repeating local structures. I'll show you a picture in a second. These are held together by hydrogen bonds. Tertiary structure is the folding of a single protein. This is when R group interactions occur within the amino acids, and there's a hydrophobic core, so it's usually hidden in the middle because it doesn't play well with water. And then quaternary structure is the complex protein. This is usually made of 2 or more subunits. And again, I'll show you pictures so you know what I'm talking about. The primary structure, right, this is the amino acid sequence. If all of these little dots are their own amino acid, it is just amino acid, amino acid, amino acid, held together by a peptide bond. There are an infinite possibility of polypeptides because there are 20 different amino acids. It could be in any length or in any order. So there's just an unlimited possibility because you could have a 3-length polypeptide or you could have a 3,000 length polypeptide. Amino acids themselves have unique properties. Again, you do not have to memorize all 20 amino acids, but you should know these properties. Some are polar and some are nonpolar. So some like water, some don't like water. Some are positively charged, some are negatively charged, and some contain sulfur. What these properties do really determines how the polypeptide is going to fold up into a protein, because it will connect to things that it likes and avoid things that it doesn't like. Okay? So for the secondary structure of a protein, you have repeating local structures held together by hydrogen bonds. Okay, so in this case, you have your alpha helix and your beta pleated sheets. All of these are like amino acids going up and around, but they're held together by hydrogen bonds, so they're forming this like sort of ringed structure or this sheeted structure. Like they're connected by these hydrogen bonds, so it's keeping them together, but it's not really like a 3D structure yet. It's sort of still, I mean it is, but it's not as 3D as what we're going to see. It's sort of like a basic new structure. Tertiary, that's when we start getting into like the proteins that we're used to seeing when we look up an actual protein. So this is the folding of a single protein into a 3D structure. This is like really what you think of typically when you think of a protein. This has a hydrophobic core, so in the middle of all of this you have amino acids that do not like water. And the folding gives proteins their functional properties, right? Like active sites on enzymes, because how it's folded determines what can interact with it on the outside. Those R group interactions, remember R groups are part of amino acids. If you have a hydrophilic polar amino acid, that's going to orient itself to the outside because it will play well with water, whereas the hydrophobic non-polar amino acids will be protected on the inside. Opposite charged ions attract each other, so they will meet up and help fold this protein into the shape that it wants to be in, and disulfide bridges form between sulfur-containing amino acids. So if you have 2 amino acids that have sulfur in them, they will connect together because they will form a bridge. So let's practice that. Let's just say I have serine. I have 11 of them. They are polar. They play well with water. All of these blue circles are polar. I have a lysine and an asparagine, so I have one yellow, one green. The lysine is positive, the asparagine is negative. I have cysteine, which contains sulfur. I have two of them, but look, they're pretty far apart from each other. And then I have valine. I have two of them. The red ones, they are nonpolar. So we know right off the bat that these valines are going to be in the middle. However this protein folds up, these valines are going to be in the middle. We also know that because I have a positive and a negative, they're going to want to meet up too because they are opposites, so they are attracted to each other. And my two sulfur-containing cysteines are going to want to meet up with each other too and form a disulfide bridge. So really we're just looking at this and thinking to ourselves, okay, how can this fold so that the green and yellow are touching, the two pinks are touching, and the blue surrounds this red? That's what you want to do when you're modeling protein folding. You have to figure out what the properties are and then figure out how they're going to actually fold up together. In this case, it's going to look like this. We knew that the reds had to be in the middle, right? We knew that the two pinks had to have that disulfide bridge between them, and we knew that the positive and negative had to line up with each other because they like to be next to each other because they're attracted to each other. We hid away the hydrophobic, and the hydrophilic are surrounding the outside, allowing this folded protein to work properly. Now our quaternary structure is when we take like two or more tertiary structures and shove them together. That protein complex, like I said, is made up of two or more subunits. You can see that this would be like one subunit. This dark blue, light blue would be a second, and green would be a third. Some examples of quaternary protein structures are DNA polymerase and hemoglobin. And then types of proteins. There are two types, and you should be able to tell me the difference between fibrous proteins and globular proteins. They're pretty much opposite of each other, but still you should be able to tell me. So fibrous are insoluble in water. That means that they're not going to really break down in water. Globular proteins can be soluble. Fibrous proteins are for structural reasons, for support, for strength. Some examples are keratin in your hair and nails, elastin in your skin, and collagen in your tissues. Some globular proteins, they're more functional, right? They're like enzymes, antibodies. Some examples of those include amylase, which helps digest starch, insulin for blood sugar regulation, hemoglobin, immunoglobulins, which are antibodies, and for transport, like sodium-potassium pumps. And then finally, we are talking about polar and nonpolar amino acids. In this case, both polar and nonpolar amino acids are used in membrane channels. Polar sections allow ions through. They're positioned on the outside surfaces and help with facilitated diffusion. Nonpolar sections bond with hydrophobic tails, which makes sense. Polar is like almost like a more friendly version of nonpolar. Nonpolar is always going to want to be hidden away from the outside. So polar amino acids and enzymes help bind the substrates for reaction, and nonpolar amino acids help bond proteins to the plasma membrane. Okay, so polar is for enzymes, and nonpolar is really like the connecting of the proteins to the membrane. Okay, so that was a quick review of proteins. Hopefully you remember some of that. If not, hopefully it wasn't too much. Either way, I'm proud of you, and I'll see you in class.